Now we move straight to the session that will be, that is going to be delivered by Dr. Sudip Mukherjee. And before we start, I'd like to introduce Dr. Sudip Mukherjee to our audience. He happens to be an MRSC, a postdoctoral research associate at Rice University and director at Avatar Biopharma, CSIR Indian Institute of Chemical Technology, Avatar Biopharma. Now, Dr. Sudip Mukherjee earned his PhD as a CSIR fellow at CSIR IICT, Hyderabad, under the guidance of Dr. Chittaranjan Patra. Currently, he's working as postdoctoral associate at Department of Bioengineering at Rice University, USA, in the supervision of Dr. Omid Visa. His research is involved in the design and development of advanced novel nanomaterials for drug delivery in cancer diagnostics, in modulatory applications, and angiogenesis. He is a member and reviewer of MSSC and reviewer of RSC United Kingdom. So we are indeed very fortunate to have gentlemen of this stature in our midst, and I sincerely believe that the audience will benefit a lot today from whatever he tells us about how to survive and survive well this curse called COVID, this curse on the whole human civilization, and how to survive quite successfully in it. So welcome, Dr. Sudip Mukherjee, and it's now over to you, sir. Dr. Sudip Mukherjee, I hand over the microphone to you.
Hello, everyone. It is indeed my pleasure to join today from the invitation of JIT. And I remember I gave the exact talk almost like a year back, if I'm not wrong, maybe like last May. So I feel like the time flies so fast, but nothing has changed. The only thing has changed, like the vaccination came in the market, and many people, I'm not sure how many of you already got, but yeah, luckily I got the vaccination. So my presentation from last time will be almost the same, because I feel like there is not much which has been changed from there. Only few things I'd like to highlight here is like few mutated strains and what type of vaccines came in the market. So let's start with a short presentation, and I also have some videos. I feel like that would be helpful for everyone to see that video. And I would like to have this more an engaging one. So please feel free to ask lots of questions, because the students mostly, they want to know. And if you have a lot of queries, please feel free to ask me. So I'll go through directly with a short presentation before. Hopefully everyone can see my presentation slides.
Yes, we can.
Yeah. So as ma'am mentioned, today's topic is on the emergence and the development of the vaccination, and how the resurgence again happened, and what type of precautions we can take to avoid further transmission. So before going to COVID, I would briefly like to highlight my current research work. I feel some of you may feel interested. So as in the introduction, you mentioned rightfully so, I work mostly on the nanomaterials and the biomaterials in the field of cell therapy in cancer and also in diabetes. So instead of chemical toxic drugs, we mostly use like human cells as a therapeutic module, and we implant those cells using like biomaterials in the body. And our work evolves not only like in cells, we do like a lot of mice study, even in the non-human primate, like in monkey study. So as you can see, like this is just a brief slide to show that how like our therapies cure the tumor. In the right corner, you can see like the untreated tumor, and then once we use our cell-based therapeutic module, the tumor growth becomes much smaller. And the advantageous point of this cell therapy is like these are much less toxic, because these are not like chemical drugs. So these are more biologically compatible to the human body. So currently my work is revolving around like mostly this type of like novel therapeutic work. And then now let's start with COVID. So everyone like COVID is like SARS-based virus, and it transmit through like cough or like sneezing. Then people take like lot of precautions, like maintaining distance or like using mask, but still we are unable to control the transmission. The only main reason I feel is like in few cases, in the few places there is like the population density is too high, and people are getting more and more casual. So as ma'am previously mentioned, like while she was traveling, she saw like majority of at least more than 50% of the people are not wearing masks. So this is really an alarming situation when people are neglecting after a certain point of time, and then the government regulation and other regulation also has come down a lot. So we don't want another situation of a lockdown, as she rightly mentioned that another lockdown can cause us severity in economically as well as like in other aspects of the society. So we must keep on following the similar regulation, even though we got vaccine. So that is the main point. Like even though we got the vaccine, we still should maintain our normal basic precautions with like wearing mask and like washing our hands, either with soap or even with the sanitizers. The main challenge from last year is like although we have vaccination, there are few mutations because these are very primary things. Mutation of any type of viruses are very common. So if you see like for normal flu virus or for normal seasonal virus, they also change, they also mutate themselves like regularly in every year. So for this COVID virus also, there are a lot of mutated strains already started coming in location wise. As ma'am mentioned before, like in India, we have a certain COVID strain, and even in UK and South Africa and United States, there are different type of mutation has already started. And yesterday was only I reading like there are double mutated virus also. So like already there are multiple mutation which has started. So, but yeah, nothing to be fear of because all these vaccinations may still work like 90 to 95% will in the body, because the major component of the vaccines are still same. So I'll go through briefly with that and how the development of vaccines happen. So in this slide, this is very old slide, and you know like lot of different companies have made vaccines already. In India especially, there are two major vaccines, namely like Covishield and Covaxin. So the Covishield, I think if I'm not wrong, it is the collaboration between the Oxford and the Serum Institute of Biotechnology, and they are mainly using the viral vector-based technology where in the small scale they are injecting a protein or like a RNA which can generate like spike protein based signal, and in a long run it will create like an antibody to your body, and so then if any virus related contamination happens, that antibody will destroy those outcoming viruses. And then another technology which is the Covaxin, that is the indigenous technology of Bharat Biotech. They use like the inactivated virus, which is although like the primary technology previously before these people used to use like the inactivated virus for vaccine. But the only one small challenge I feel is like if the virus gets mutated a lot, then this technology may not work well because in these cases you only inject that particular virus either in a very low dose or like in an inactivated form. So if there is multiple mutation starts, there are always like risk of again getting contamination. So to avoid that, scientists may need to like again remake those type of particular vaccines, especially with the Covaxin I'm talking about. And then in United States, we are mainly having two type of company which are giving right now the vaccines. Is one is from Moderna Bioscience, and then one is from Pfizer, and both of them are following like the mRNA based technology. So as all of you know, like mRNA or DNA, these are like small component of the cells. So instead of the whole virus or instead of the inactivated virus, they are just taking like a major component of the virus gene, so that if the virus gets mutated also, that particular commonality will still be same, and it will create like the prevention in a better way for long run. So I got the Pfizer vaccine, and both it is like the mRNA based technology. And although these are not like 100%, like they can't prevent you 100% from the vaccine virus contamination, but at least the papers what have been published is like they can prevent almost 90 to 95%. So still there is a chance of getting contamination even though you have the vaccine. It may not affect you in a bad way. If you didn't have the vaccine, it may like cause you more harm to you. So once you have the vaccine, the symptoms will be anyway very mild, and you may recover also very fast. So that is the only advantage to have the vaccine. So please, whenever you get a chance, take the vaccine. I know like especially the young population like us are the last to get the vaccine. First it will be like the health worker and the primary worker who will get the vaccine, followed by the old people, and then I think the young people. But I am encouraging everyone, whenever you get a chance, take the vaccine without like any hesitation in your mind, because although there are very few cases of like having like fever or blood coagulation, but don't pay that much of notice. If you have like any heart related problem or diabetic related problem, then of course consult to your doctor before taking the vaccine. Otherwise I feel like it is completely safe, because the safety has been tested not only like in various model, like it has been tested directly on human and like thousands of human patients. So the safety are already, the safety profile is established. So don't keep any hesitation and take the vaccine whenever you get a chance. And then this slide is like for the therapeutic advancement what we mostly have. So when I presented last year, I remember like I highlighted quite a few drugs starting from like remdesivir and chloroquine. Even that time I remember like people were fascinated to use like chloroquine, and a lot of people were taking that without like any consultation, and even though that was not that effective, like people were taking without any recommendation. But mostly in United States right now, they have approved, FDA has approved the remdesivir drug in case of any viral contamination happens from the COVID. So I need not to say too much about this drug, but I can say like this will be only used when the viral load is very high in your body and you are like suffering from severe mild to like severe contaminations. So otherwise, doctor always recommend to take a just normal approach where you stay in your home and see like how that goes for a few days. And if there is not much of contamination, then you don't take any medicine. Like in case of severe lung inflammation or severe pneumonia only, then they recommend for this type of drugs, because these drugs are also not like biocompatible drugs. Like these are all chemical drugs, as I mentioned before. And another therapeutic approach I wanted to highlight is like the plasma based therapy. This is also FDA has given like an emergency approval, although it is not like FDA approved drug, but FDA has given like emergency approval. So many of you have heard this. So plasma is like nothing but like a more major part of your blood where it has like all the therapeutic enzymes, antibodies, and water. So the patients who already got COVID, they develop the antibodies against the COVID after a certain period of time. It may take like a week, it may take like 2 weeks to develop those antibodies. And doctor highly encourage those who recovered already from COVID, they donate their plasma, because with their plasma, they can have like huge amount of anti-COVID antibodies. And if you infuse or inject that antibodies to a COVID patient, they get like a kind of an external force to protect, like to fight against those virus contamination. So this is also another form of therapeutic modality people are using apart from like chemical drugs. But again, these are only for when if you have like very severe kind of contaminations. Otherwise, in case of asymptomatic or like very low symptoms, doctor recommend you to stay as much as possible in your home. And then in case of severe pneumonia, you may go and take this type of treatments. Uh, so what is the aftermath? Because there are multiple mutations already started, and the challenge, as previously I mentioned and madam also mentioned, like people have stopped taking any type of precautions. So uh, everyone has to understand here, like this is not something like a seasonal disease or like any disease which you can prevent by taking one vaccine. These are like uh, normal flu type of vaccines, like flu type of disease which will stay with us forever, because virus, especially this type of virus, will keep on mutating themselves, and scientists may have to like prepare vaccine every year. So I'm not sure right now if we need to take the vaccines maybe like alternative 2 year or 3 year time period, but at this point we don't know like how it will be in next 3 years. So I feel after COVID, the whole geographical and the social structure has completely changed, and we need to be more responsible towards not only towards our family, towards our society. And unless this type of transmission completely like get down to like very negligible level, we should still keep on maintaining the social distancing as much as possible. I know like all the school colleges have already started, and we can't prevent that because the economy has to keep rolling. But at least what we can do is like still we can have our normal precautions with mask and social distance, along with like our own hygiene protection. Keep on using like soap and water. I prefer soap more than like sanitizer, because sanitizer can also kill your good bacteria, and that can also have like a long term effect, because it is kind of a symbiosis between like human body and bacteria virus. Like you can't actually prevent virus contamination and bacteria contamination completely, because if you see microscopically, there are maybe like thousands and lakhs of virus and bacteria roaming around our body. So we have to live with them, we have to like create a symbiosis with them, where we must prevent the bad bacteria and virus, but still we should live with those good bacteria or like relatively less harmful bacteria and viruses. So yeah, as I mentioned, keep on maintaining the distance, and don't be scared about the mutations and all, because there are, although multiple version of mutated virus already came, but these vaccines which already are in effect, they are also showing at least like 80 to 90% of successful prevention. And even like few days back, like I remember the Argentinian president, he got the COVID infection even though he had the vaccines. But as I mentioned, like if you have the vaccine, the contamination will be much less, and it will cause much less, like it will cause less harm to your body. So after these, I would like to show the video which I felt can be helpful for to understand the whole mechanism, the disease mechanism. So I'll straight go to that video. while it is getting downloaded, I would encourage anyone, if you have any question, just let me know, because it would be better if we can keep on discussing on various confusion if you have, or various query you have, and then we can start this video. So yeah, feel free to ask any question in the meantime.
Any questions from the audience?
This is SARS-CoV-2. It belongs to the family of coronaviruses, named for the crown-like spikes on their surfaces. SARS-CoV-2 can cause COVID-19, a contagious viral infection that attacks primarily your throat and lungs. What actually happens in your body when you contract the coronavirus? What exactly causes your body to develop pneumonia? How would a vaccine work? The coronavirus must infect living cells in order to reproduce. Let's have a closer look. Inside the virus, genetic material contains the information to make more copies of itself. A protein shell provides a hard protective enclosure for the genetic material as the virus travels between the people it infects. An outer envelope allows the virus to infect cells by merging with the cell's outer membrane. Projecting from the envelope are spikes of protein molecules. Both a typical influenza virus and the new coronavirus use their spikes like a key to get inside a cell in your body, where it takes over the cell's internal machinery, repurposing it to build the components of new viruses. When an infected person talks, coughs, or sneezes, droplets carrying the virus may land in your mouth or nose and then move into your lungs. Once inside your body, the virus comes in contact with cells in your throat, nose, or lungs. One spike on the virus inserts into a receptor molecule on your healthy cell membrane like a key in a lock. This action allows the virus to get inside your cell. A typical flu virus would travel inside a sac made from your cell membrane to your cell's nucleus that houses all its genetic material. The coronavirus, on the other hand, doesn't need to enter the host cell nucleus. It can directly access parts of the host cell called ribosomes. Ribosomes use genetic information from the virus to make viral proteins, such as the spikes on the virus's surface. A packaging structure in your cell then carries the spikes in vesicles, which merge with your cell's outer layer, the cell membrane. All the parts needed to create a new virus gathered just beneath your cell's membrane. Then, a new virus begins to bud off from the cell's membrane. Now, with the virus spreading in your body, how can you develop pneumonia symptoms? For this, we'll have to look into your lungs. Each lung has separate sections called lobes. Normally, as you breathe, air moves freely through your trachea or windpipe, then through large tubes called bronchi, through smaller tubes called bronchioles, and finally into tiny sacs called alveoli. Your airways and alveoli are flexible and springy. When you breathe in, each air sac inflates like a small balloon, and when you exhale, the sacs deflate. Small blood vessels called capillaries surround your alveoli. Oxygen from the air you breathe passes into your capillaries, and then carbon dioxide from your body passes out of your capillaries into your alveoli so that your lungs can get rid of it when you exhale. Your airways catch most germs in the mucus that lines your trachea, bronchi, and bronchioles. bronchioles. In a healthy body, hair-like cilia lining the tubes constantly push the mucus and germs out of your airways, where you might expel them by coughing. Normally, cells of your immune system attack viruses and germs that make it past your mucus and cilia and enter your alveoli. However, if your immune system is weakened, like in the case of a coronavirus infection, the virus can overwhelm your immune cells, and your bronchioles and alveoli become inflamed as your immune system attacks the multiplying viruses. The inflammation can cause your alveoli to fill with fluid, making it difficult for your body to get the oxygen it needs. You could develop lobar pneumonia, where one lobe of your lungs is affected, or you could have bronchopneumonia that affects many areas of both lungs. Pneumonia may cause difficulty breathing, chest pain, coughing, fever and chills, confusion, headache, muscle pain, and fatigue. It can also lead to more serious complications. Respiratory failure occurs when your breathing becomes so difficult that you need a machine called a ventilator to help you breathe. These are the machines that save lives and that medical device companies currently ramp up production for. Whether you would develop these symptoms depends on a lot of factors, such as your age and whether you already have an existing condition. While this all sounds scary, the push to develop a coronavirus vaccine is moving at high speed. Studies of other coronaviruses led most researchers to assume that people who have recovered from a SARS-CoV-2 infection could be protected from reinfection for a period of time. But that assumption needs to be backed by empirical evidence, and some studies suggest otherwise. There are several different approaches for a potential vaccine against the coronavirus. The basic idea is that you would get a shot that contains faint versions of the virus. The vaccine would expose your body to a version of the virus that is too weak to cause infection, but just strong enough to stimulate an immune response. Within a few weeks, cells in your immune system would make markers called antibodies, which would be specific for only the coronavirus, or specifically its spike protein. Antibodies then attach to the virus and prevent it from attaching to your cells. Your immune system then responds to signals from the antibodies by consuming and destroying the clumps of viruses. If you then catch the real virus at a later stage, your body would recognize it and destroy it. In other words, your immune system is now primed. Collecting evidence on whether this will be possible, safe, and effective is part of what's taking researchers so long to develop a vaccine. It's a race against time to develop a vaccine amid a pandemic. Each step.
Okay, so the key takeaway from this video, as I already mentioned, I think you understood like how the virus contamination can cause, and then it mostly affects like your lung cells, and further the development of vaccines using the inactivated virus or the mRNA-based technology. So yeah, this is overall, like I think everyone mostly knows about COVID in last 1 year, because we already completed like over a year and almost half. So everyone knows about vaccine, everyone knows about the precautions, but it would be better, I think, if we can discuss more through the question and answer, then it would be like more helpful for everybody, I feel.
We are expecting questions from the audience. Anyone has questions to put to Dr. Mukherjee?
Hello.
Yes, please. Anyone wants to question?
Hello, and Dr. Sudip Mukherjee, myself, Gopal Chakraborty. I want to ask a question to you that I have a doubt that you know that many movies was released far before this COVID-19. Okay, like say Pandemic, Contagion, which was released like 2010, 2011. Okay, so which are almost similar to like this pandemic. Okay, that we have found. So what researcher thinks today, after completion of 1 year, that is it really a human made in research lab, or is there any update related to this?
So that is it is a good question, obviously, but that is a controversial point. Like being a researcher, our job is not really to look in that aspect, but to see like how it came, right? So I feel there are like multiple thoughts on how this specific like strain came. One can be because this SARS is not a new virus. This already has been there, and there has been like some similarities of this strain with like previously identified strain. So this was already there, and maybe through like consumption of like zootonic species or like that kind of animal consumption, maybe it got contaminated from like bat or like from snake to human. But there are other thoughts also from a certain part of society who feel like it is lab made, but nothing has been proved yet. So in that case, I would uh, like feel like this is just kind of like a normal virus which like generated over the time period, because with time this environmental landscape is like changing rapidly, and as like the pollution is increasing with like decreases of like plants and like increasing of human habitat, more and more these virus and bacteria will start like affecting us, because you know like the global warming also, and as the ice is melting, there are a lot of like virus and bacteria which were there inside of those ice, like kind of anthrax. We have never heard of this anthrax last like couple of years, but with ice melting, this type of old viruses or like old bacteria, they're also coming out from like deep of the earth or like deep of those ice area. So yeah, like that is my takeaway, I guess. It is difficult to say at this point if it is like lab made, but as you mentioned, there are people and community or like scientific community, even from the government, who they believe like it is probably like lab made virus, but nothing conclusive yet.
Thank you, Dr. Mukherjee.
I think our colleague Amrita has a question. Let's see what it is. She has probably written it
Yeah,
out here.
I can see the question. Yeah, how quickly could COVID-19 vaccine stop the pandemic? So I think you have to see this as a individual wise. So once you get the vaccine, it takes about like 7 to 14 days. So I didn't mention like the vaccine dose, how they are giving. So let me little extend that. So for any vaccine, they are nowadays giving like 2 shots. The first one is with little low dose, just in order to like give your body little more time to adjust with the vaccine, and then they are giving like the second dose in like 3 to 4 weeks apart from the first dose, and the second dose is much more heavy dose than compared to the first dose. So once you get both the dose, the first dose itself may not work well. So once you completed your full dose, then you expect like at least another 7 to 14 days to give your body that period of time to generate enough sufficient amount of antibody against the COVID. So it is kind of just like a memory, right? So you inject those mRNA or small scale of inactivated virus, and your body will get that memory and create like antibodies against them. So individual wise, it takes at least like 1 month for you to get protected. And now to stop the pandemic, it is a very difficult question, right? Because to stop the pandemic, then you have to vaccinate everybody. So a country like India, where we have like almost 1.3 billion people, it is not easy to vaccinate, because in a rapid scale also, if we vaccinate, it may take at least 1.5 years to 2 years to completely vaccinate everybody. So I still feel like if like a certain number of people in the society get vaccinated, but others, if they still maintain these precautions and still if they maintain this urgency uh, against the disease, because if they get reluctant, if they get casual, uh, what is like with second wave, like fourth wave, fifth wave also can come. Like there is nothing called second wave, it is a continuous process. So uh, as much as fast we can vaccinate everybody, like we have to hope, like but yeah, things are getting better. But as in India, all you know, like the second waves are starting to come, so still there is concerns. So as I mentioned, I suggest to keep the precautions. Any other question?
Yes, I'd like to ask you one question.
Please.
It won't be like I won't be requesting you to answer the question, answer any controversial question, but I'd like to put this question put by Gopal Chakraborty in a slightly different manner. It is
Sure.
it is simply out of my curiosity as a layman who hasn't read medicines. My question is, I am not asking you whether this COVID-19 and or this particular virus was designed in the laboratory or not. My question is it at all possible to design such a virus or create such a virus or culture such a virus in a lab? Is it possible?
Yeah, it is absolutely possible. Like people do virus culture all the time, because this use of vaccines, like they also use a viral vector. So viral vector is nothing but like a uh, non-harmful virus, right? So you have to anyway culture virus for the vaccine also. So you can culture virus,
Okay.
either it can be non-pathogenic or can be pathogenic. But people like viral labs also have to culture the pathogen virus in order to understand the disease mechanism, in order to like create the vaccine. So it is absolutely possible to mutate the virus also, because you can genetically infuse or transmit some type of like genetic material like mRNA or like other type of plasmid DNA to any type of cells or virus to change their basic structure. So yeah, like in lab scale you can certainly make, but I'm still not sure like if this is uh, made in the lab or not, but you can do that.
Okay, sir. Thank you again on behalf of all the organizers for sharing this valuable time with us. And I'm sure that everyone has benefited a lot from whatever you told us about this great threat to human civilization, COVID-19.